We all carry about 10 times as many bacteria in archaea as our own cells. Microbiomes are microorganisms such as bacteria, viruses, or fungi found in a particular environment such as the human body and digestive tract. The increase in bacteria in microbiomes of different species have shown to provide several benefits to one's environment and human health.
Human health has been significantly impacted by the presence of microbes in their gut. This impact has been both negative and positive for human health. Also, since microbiomes are unique to organisms, they can be used to biodetect the source of fecal contamination.
These microbial communities have critical roles in genetic and metabolic attributes of the host organism. For instance, the gut microbiota has provided humans with the ability to break down previously indigestible polysaccharides such as cellulose and resistant starches. In addition, it regulates the host immune system and protects the body from pathogens. In both animals and humans, changes in the gut microbiota are related to diseases such as Crohn's disease and ulcerative colitis. Due to adaptations based upon the host's health and microorganisms' lifespan, the composition of these communities differ in humans, vertebrae, and other animals. For example, there is evidence of how social behavior affects the gut microbiota of bees. The emergence of the corbiculate bees from solitary ancestors coincide with the 5 core gut bacterial lineages. This is a mutualistic relationship that provides the host with strong gut integrity and energy as the microbes gain glycans in a protected anoxic environment.
The diversity of the gut microbiomes in the GI tract is due to coevolution and host selection. Research shows that the Cytophaga Flavobacterium bacteroids division is mostly derived from mammals and has gone through evolution after adapting several mutualistic relationships. In addition, the development of the gut microbiome begins at birth and progressively evolves due to the presence of illnesses and changes in diet. This contributes to an increase in the diversity of a microbiota over time. With that, the structure of the microbiome reflects natural selection in 2 ways. One is at a microbial level, where survival strategies impact the fitness of the bacteria, and the other is at a host level, where functionality is reduced in the microbiome and results in a decrease of the host fitness. Microbial consortia, which is when 2 microbial groups can live symbiotically, can have different effects on the host fitness. The activities of the microbial consortia can reduce the host fitness, which leads to the loss of these hosts. However, the microbial consortia can also promote host fitness, which creates more habitats for hosts.
In humans, microbiota are known to strengthen the integrity of our intestine, boost our immune systems, and aid in digestion and nutrient uptake. Without them, the human body is vulnerable to gastrointestinal diseases and harmful bacteria like Clostridium difficile, which is known to cause diarrhea and severe abdominal pain. Speaking of C. diff, the treatment for those infected by that bacterium or those who are suffering from a depleted microbiome, a procedure called fecal transplantation, is evidence of the importance of the human microbiome. And yes, fecal transplantation is exactly what it sounds like. Doctors at Emory School of Medicine have nearly perfected a technique to transplant healthy feces from a donor into the colon of someone else to rebuild their microbiome. Now, studies are being conducted to connect the health of the human microbiome to the health outcomes and disparities. For example, researchers at the Virginia Commonwealth University investigated the factors that impact the microbiome of African American women during pregnancy and determined that a weak microbiome can be connected to premature birth.
Since every organism has a unique composition of gut microbiota, the specific 16S RNA sequences of bacteria in fecal samples can be attributed to specific animal species. This is especially important when it comes to the fecal contamination of freshwater sources. The presence of avian feces in water sources leads to the formation of microorganisms that negatively impact health for both domestic poultry, shellfish, and humans. Understanding which organisms are responsible for the contamination of a specific water source can lead to solutions that provide clean water. Also, understanding which organisms have contaminated the freshwater source can help scientists determine the impact certain animal feces actually have on human health. For example, studies have proven that avian fecal contamination of water is not as harmful to human health as human fecal contamination of water. This is because most avian parasites cannot be cross-contaminated. That is, they have adapted to exist only within their specific birds.